Hi, hello. Let's start. It's my pleasure to introduce you to our man from Chicago, Dr. Professor Aaron Landay. He's from the Department of Internal Medicine from the Rush University, and he's going to presenting HIV and aging role of the microbiome. Thank you very much for it.
Bom dia. I'll give you my only Portuguese for the day, and I will speak in English, as my other colleagues today. Thank you again, and as I think everybody has said, a little feedback. I'd like to also give my greatest thanks to Esper and the organizer, Esper Callis. And I think Esper demands a round of applause for everything that he's done. This is a wonderful meeting that I always look forward to coming to Brazil for, and it is great, and it's great to be with all of you again and hopefully provide some new information in HIV. What I'd like to do today is really show you about the intersection of HIV and aging, which is a critical area for all of us. We're all getting older, and I myself have moved to the section of geriatrics in the Department of Internal Medicine to now work more in the aging interface with immunology. And I want to introduce some newer concepts to you today about the aspects of health and longevity. We're now looking at the aspects not just of lifespan, but we're replacing that now with health span. And there's a new field that you may not be familiar with that actually at the National Institutes of Health has been developed across all of 27 institutes. It's the field of geroscience, or the science of aging, or gerontology. And we're moving now to a consideration of the span of longevity to consideration of health span. There are actually now meetings at the NIH, there will be one this fall, on the concept of geroscience. HIV has provided a lot of information and really contributes greatly to this new field of aging. And we're looking at domains used to characterize health span. And many of these are the outcomes of the non-AIDS outcomes of neuromuscular function. We've heard a bit about cognitive. We'll hear more about cardiovascular function later, metabolic, and inflammation or inflammaging, and then finally body composition. And then there's a shift towards delaying and compressing the period of the lifespan when frailty and disability increase, and thereby extending and improving your health span. And a lot of this is what you're seeing in your HIV patients. This aspect of they're becoming more frail, there's more disability, and we hope then with some of the things we can learn from the fields of aging now, actually implement those into the fields of HIV. So we're really crossing borders here in the both areas. I mentioned a new concept that some of you may or may not be familiar with, that's inflammaging. Again, inflammation as it occurs in the context of aging. There's an increase in the number of pro-inflammatory molecules in the circulation. It's associated with this change and dysfunction of these components. And many of these inflammatory components, and I'll talk more about them, are the same inflammatory mediators we study in HIV, IL-6, TNF-alpha, IL-1. All of these are the same components that the aging field has been studying now for 20 or 30 years. But there's been very little dialogue between the two fields to understand what goes on in aging is also happening in HIV in a potentially accelerated phase, especially in inflammation. And this elevated inflammatory state that occurs triggers or facilitates many of the age-related diseases, and those are the same ones listed here, such as cancer, cardiovascular disease, neurocognitive, metabolic diseases. These are all the non-communicable diseases we've also learned about in HIV. What I'm going to do today is to focus on one aspect of what my lab has been doing over the last 10 years and intersecting between aging and HIV, and that's looking at the gut microbiome. There's a lot of interest now in the microbiome in aging and many fields of medicine. This has gotten a lot of interest now, and actually one of the centers that was the National Genome Center at NIH, where the human genome was first cloned by when Francis Collins was the head of that center, has moved on to studying the human microbiome. And there are now global efforts to understand more about the human microbiome and how it impacts health and disease. And there are many national and international meetings on this. Again, what we know here is that in numerous immune-related disorders are altered by the microbiome. Much of it is looking at the diversity, and this is known as alpha or beta diversity. So in the gut microbiome, you want a great deal of diversity. So you want a lot of diverse bacteria present there. Interestingly, in other microbiomes on the host, and there are many, and I'll talk mostly today about the gut, in the vaginal microbiome, you want a more restricted diversity. So homogeneity is healthy, whereas when you get diversity, as in bacterial vaginosis, you get other effects and negative outcomes. There's also the idea that there are fewer butyrate-producing bacteria and more gram-negative pathobionts are prevalent during aging. And some of these changes also are occurring, as you'll see in a moment, in HIV as well. Butyrate is an important mediator. It's a short-chain fatty acid made by bacteria in your large intestine. It is anti-inflammatory in nature, and it has an important role in maintaining the gut barrier function and gut barrier integrity in health. And then when you lose these bacteria, your gut becomes leaky, both in aging and in HIV. So the various metabolites, as I've just said, affect the mucosa and peripheral tissues and influence the overall host metabolism. So when you lose butyrate-producing bacteria, you alter both your fat and your glucose metabolism. So this leads to many of the aging effects of metabolic outcomes. Again, there's alterations in microbial populations, changes in gut permeability, and diffusion of LPS into the circulation. There's also a link between the gut microbiota and host behavior and gut-brain signaling. So there's an important role for how the gut may actually affect the brain and brain function during aging and leading to neurocognitive changes in aging, including Alzheimer's disease. There was last year a whole 2-day meeting on the microbiome and Alzheimer's disease, where investigators from all over the world came together to talk about this, how the gut microbiome through diet and other alterations can affect Alzheimer's. So I think you're going to find that by studying the microbiome, whatever field you're in, you'll learn a lot about biology. This is just some data that we generated a number of years ago, looking at young individuals at 30 years and elderly at 75. These are all healthy individuals, and we found that there is, again, a significant increase in LPS levels during aging. That's healthy aging. Over age 70, you find elevations in microbial translocation. When we then look at HIV in the gut microbiome, again, many of the same changes occur as we saw in aging. We get a decrease in gut diversity of the alpha and beta diversity. The taxa composition and metabolism have been, again, are different. The normal microbiome also, which is what we see in our healthy controls when we look at comparators, are not restored during antiviral therapy. So there are still consistent changes in the microbiome with even with therapy and treatment early. This has been shown even in the earliest studies. Irene Soretti's group at the NIH has studied this in cohorts from Thailand of acute infection and shown that they still have and maintain, even in the earliest treated patients, alterations in the gut microbiome. There is, again, shifts in Prevotella. Prevotella is a normal host commensal found in your gut. And what happens during HIV and aging, these Prevotella become pathobionts. They actually take on a role and create an inflammatory environment in the gut in our HIV patients. And we lose the Bacteroides species of bacteria that are important in making short-chain fatty acids. And so this is where we then also find these changes. There's also changes that are associated with systemic inflammation and translocation. So I'll talk a bit more about that. But more importantly, it's not just who's there in terms of the bacteria, it's what they're doing. And I'll get more into the aspects of the metabolomics of the bacteria themselves are more critical than just quantitating the bacteria themselves. And then finally, one of the important variables that we have to think about is the dietary associations. As we're looking at a global population, diet has a significant effect on your microbiome. And any studies that you're doing here in Brazil or anywhere in the world, taking a diet history is critical. One of the areas we've begun to collaborate on with Mohammed Abdelmonsen at Wistar Institute is looking at the gut glycome. This is looking at the actual sugar and sialic residues on cells and bacteria in the gut. And we're finding now that the gut glycome is important in terms of the homeostatic relationship between the gut and the microbiota. And so we're now looking at this idea of the glycome structures on the epithelial cells, that there is important role now for the bacteria that can degrade and regulate now the whole glycome and affect our outcomes. We've begun to look at this and did a pilot study here in patients that we had seen at Rush who are HIV positive. They're mostly males. They have a median age of 50. They're virologically suppressed with less than 40 copies, and they have good immune reconstitution at over 500 CD4 cells. And what we did then do here with Mohammed's lab is look at a large array of the glycome markers in the blood and plasma in terms of the circulating glycome profiling, but more importantly, we had samples across the GI tract, and you heard from Lish in his last talk the important role of looking across the entire GI tract is critical in terms of finding differences of whether you're looking in the right colon, the sigmoid colon, or the ileum. And he was showing you a lot of the action he found was in the ileum, where we also find differences. We then looked at the microbiome by 16S sequencing and the glycome profiling, HIV DNA quantitation, and now we're also doing targeted PCR on genes across the different profiling. And what we found, interestingly enough, is that the sialic acid catabolism may play an important role in the microbial composition, that there's a shift, importantly, in the microbiome during HIV in terms of release of sialidase, and the sialic acid catabolism occurs. And so the bacteria that are then present and remain in the gut, which are different in the HIV positive versus our negative controls, was that we now find hyposialated presence of the actual glycome, and this is linked directly then to the particular bacterial species. I won't go into the details here, but we're now finding those differences are linked to the sialic acid, loss of sialic acids on our proteins present in the gut compared to the bacterial species. We can actually show then that these mucosal sialic acid catabolism and also fucosylation may play a role in the composition and shift in our ART-suppressed patients. So we then find that in hyposalated in the gut, the hyposalated presence of these from the gut actually links to the plasma levels of changes in important mediators, such as soluble 163. When you find higher sialate present proteins, you then find the lower levels of soluble 163. And then also we lose the fucosylated glycans. Higher fucosylation also links to lower soluble 163. So we're now finding linkages in terms of the soluble inflammatory mediators and the glycosylation changes directly in the gut of our individual patients. We've also gone on in these same individuals then and looked at HIV DNA copies and find that there is now a direct correlation of hyposalation present in the gut tissue and the copies only of the ileal-associated HIV DNA. So it's a critical change in the ileum in these sialic acid changes that might be relevant then to the reservoir of HIV. Now we're looking at this further in many more patients and digging deeper in terms of the actual reservoir measurements in our patient population. So we think this is a critical area that we're beginning to study, that the gut glycome regulates the homeostasis between the gut and its microbiota. There are a number of papers from Nature and Science that have looked at sialic acid catabolism driving intestinal inflammation, and fucosylation sustains the symbiosis. And we're now doing this in monkey studies where we're actually replacing sialic acid ratios, molecules, and we're blocking the sialidase. The actual drugs that are used to treat influenza, are actually sialidase inhibitors. So we're actually treating our monkey populations with SIV infection to try to block sialidase and also give back fucose to our monkey population. So this is actually the subject of a recent grant submission to NIDDK that we're going to be continuing on looking at this role. And we also note that many of these changes, and we published this earlier, of the sialic molecules that are changed in the peripheral blood also link to the communicable diseases, neurocognitive, and we now have a grant with preliminary data on cardiovascular disease. So these new sialic molecules may give us more insights from the gut into the non-communicable diseases. This is, again, a study that was done by Carol Wilson's group and Stephanie Dillon at the University of Colorado to look at the differences in how HIV and aging affect the host microbiome. In this particular study, they're looking at people living with HIV from ages 50 to 75 on heart versus seronegative matched controls at 50 to 75, looking at stool samples, looking at microbiome, stool short-chain fatty acid, and then systemic markers, many of which I've just spoken about. And then there were clear changes in the effect of age on microbial taxa. So clearly, we're able to show directly that important changes, and many of the changes are the ones I referred to, loss of butyrate-producing bacteria that are present. There were also, in this study, looking at the effects of age among HIV patients and then age among the controls, and there clearly are differences. So there is an impact of HIV during aging beyond that just of aging effects on the host microbiome. So you really have to take into account now, as our HIV patients are aging, we're going to see an increase in the various different bacteria here that are going to have negative effects, and then that may be important in terms of the losses or the gains of different populations, and clear differences, as I mentioned before, linked to the butyrate and short-chain fatty acid producers, which is one of the main features that seems to drive, and there's an additive effect of both aging and HIV on these butyrate-producing bacteria that also lead to more inflammatory responses systemically and probably help to drive a lot of the non-communicable diseases. So what was this study show that HIV and AIDS were independently associated with distinct changes in the microbiome of older populations, that HIV infection status influenced age-related changes, so that, again, there are critical impacts of HIV itself during the aging process, and the associations between the taxane inflammatory markers, stool metabolites, and dietary factors will differ depending on the HIV status. So all of these are going to be complex interactions, and you really have to take into account all the variables that are being measured. But what is very clear is that HIV itself does have additive effects during the aging process on the host microbiome. As I alluded to, what we're interested in more is not just the quantitation of the bacteria, but more about the metabolome. And this is really an important new area of study that a lot of the groups are starting to look at and show that, as I said, short-chain fatty acids, these include both propionate, butyrate, and acetate that are produced by the gut microbiota, mostly in the large intestine. And butyrate has been shown to also reactivate latent HIV through its effects on inhibiting histone deacetylase. I'll come back to that in a moment. We're actually beginning studies with John Carn at Case Western to study this more directly. We've had some preliminary data to look at the role of butyrate, and John actually has published on this already using some of his latency models to show that if you take bacteria from various sites in the host, and actually you can show they can reactivate the virus through the presence of these short-chain fatty acids. Butyrate production also correlates with a decrease in monocyte activation, inflammation, and then the bacteria may also be important in terms of activating HIV replication. So there's important mechanism then of the bacterial-derived metabolites on the host immunity. As I said, the short-chain fatty acids are only one of many here in terms of looking at succinate amino acids and then other secondary bile acids. So there's a whole variety of these metabolites that are important and have a role in driving the biology and the immunology. So there's a critical interaction between the bacteria, their products and metabolites, and then all of the various immune cells in the gut, including both innate and adaptive immune cells overall. What we've begun to do is to actually do systemic markers and published on the correlations of these systemic markers of the bacterial metabolites and outcomes related to cardiovascular disease mainly. This was a recent paper published by our group in Journal of Infectious Disease looking at the choline metabolite TMAO and finding a highly significant association in our HIV-positive patients of increased levels of TMAO and the outcomes related to cardiovascular disease and coronary artery calcium. We also find that there are important correlates here related to activation and also the kynurenine and tryptophan ratio. So we can show that the K to T ratio systemically correlated with TMAO changes. So there's a link between many of the different metabolite changes that we've seen they're not independent variables, but many of them do show correlates together between driving T-cell activation here and soluble CD14 monocyte activation and other metabolic changes. So what we found in this particular study was that among HIV-infected participants, higher plasma TMAO was significantly associated with an increased risk of incident carotid artery plaque. This was done in 1,000 patients through the WISE and MAX cohort study. So we have good clinical data, good outcomes in these patients related to HIV-negative controls. We also found that the plasma TMAO was positively correlated with the K to T ratio, soluble serum 14, and 163, but not with traditional CVD risk factors or global inflammatory markers such as IL-6. So there seems to be, again, an important role with some of the more monocyte-driven responses here in terms of soluble 14 and 163. And also, HIV-infected participants, TMAO was associated with risk of plaque that was partially mediated by some of the other activation markers. We've also gone on to look at the lipid metabolism using ceramides, and we can also, and just recently published this paper, that plasma levels of ceramides are significantly higher. These elevated ceramide levels, and I won't go through all the details, some of them are listed here, are associated with ART use, particularly protease inhibitor use, interestingly enough. So this may be, again, the drug class is critical in how that's going to affect the microbiome outcomes, and we're beginning to look more at that. Again, the C16 and C24 are closely correlated with monocyte activation inflammation, similar to what we found with the TMAO. And then these ceramides are also associated with increased risk of carotid artery plaque. So this gives us an additional pathway that we're now pursuing in terms of studying outcomes related to cardiovascular disease. We've also gone on and looked at the association not only with cardiovascular disease, but with diabetes. And these are, again, subjects from the same cohorts I've just shown you, with and without diabetes. I won't go through all the clinical characteristics here. But what we found is the relative abundance of 4 bacterial genera were different by the diabetes status. And we can find that here in the HIV-infected, this, again, Feingoldea was a significantly elevated bacterial species in our HIV positives and versus HIV negative cohorts as well. So we're able to find differences in different bacterial species as well here in terms of Snethia being much higher and also the Anaerococcus. All of, again, these particular bacteria that are linked in our diabetes patients may be important, and we're looking further at their metagenomic profiles now. We're beginning to isolate the different bacterial species from this original paper and now go in a deeper dive in terms of their functional relevance. We've also gone on to look at tryptophan metabolism in this particular paper, again finding that the tryptophan metabolism levels are increased in diabetes, in this case both in the HIV positives and the HIV negative populations, both in terms of the kynurenine and the tryptophan versus other markers like ADMA, proline, or alanine. So the changes aren't specific to HIV, but they're consistent with what we're finding in many of our HIV studies, either with cardiovascular disease or diabetes now, that there are clear changes in the metabolic profiles that may be linked together. We've also gone on to look in these same individuals in terms of their glucose metabolism in our diabetes patients. And there's also an important role in many of these bacterial products, which we're looking at, that may be critical in driving the metabolic processes, as what we're now finding in T cells is that there's an important immunometabolic profile that we can study in our activated populations related to glucose metabolism, pyruvate, and then outcomes related to the glycolysis and various also cytokines and fatty acid metabolism. So a lot of the various metabolic products I've just shown you are important, as I told you, particularly butyrate and others, in driving these glycolytic and fatty acid metabolic processes. What we've gone and looked at are a variety of biomarkers like GLUT1 expression. We've looked at glycolysis pathways and oxidative phosphorylation, which we've done through use of the seahorse pathways. And this is, again, data from these same diabetic patients that I've just shown you that we've looked at in terms of their microbiome. We've gone on to do other immune studies to show that CD4 T cells from women from these same HIV-infected women with diabetes have increased metabolic activity based on their GLUT1 expression, and then also looking at both the oxidative phosphorylative and glycolytic pathways. These are looking at 10 patients each with purified CD4 cells in terms of using the seahorse analytic approach, so that there again is activated metabolic pathways that go along with changes directly in the microbiome, and we're now looking at the direct effects of the microbiome products on these metabolic pathways. We've also gone on through using the Fluidigm qPCR system to show that these same patients' CD4 T cells from the ones I've just shown you, elevated GLUT1 expression, heightened oxidative phosphorylate and glycolytic activity from the functional perspective, also have increased expression of metabolic genes from the glycolysis pathways, TCA cycle, and pentose phosphate shunt. I'm not going to go through all the gene pathways here, but it just to state here in the diabetes, they're significantly elevated to the patients with no diabetes. So again, both at the RNA level and the protein level, we're finding metabolic changes in our patients, and I think now these are going to allow us to look at critical pathways that we can now intervene with, which I'll show you in the rest of my talk. So the real question is, once we've identified these particular pathogenic changes in the context of the host microbiome, both in HIV and aging, what are our therapeutic options and what can we do? And there are a number of approaches we've begun to think about, including prebiotics. We've done a number of studies in probiotics, including in the ACTG. We're looking directly at postbiotics using butyrate itself to treat symbiotics, where we combine the two, and then finally there's potentially a role for fecal transplants that are also beginning to be addressed in the HIV population. In terms of the use of prebiotics, we've been working with groups at actually Purdue University, Bruce Hamaker's group, who's head of the Carbohydrate Center, Whistler Center there, who's actually producing tailor-made prebiotics. These are both soluble and insoluble fiber material that we can then use to induce different levels of short-chain fatty acid producers here in terms of making propionate, butyrate, or acetate in our in the patients themselves. We've actually looked directly in this study from some patients at Rush to see what happens in terms of just some simple questions. What does ART therapy do to the short-chain fatty acid levels in the stools of our patients? And we can find, at least overall, there's some response here, pre-therapy, 6 months and 1 year post-therapy, looking at the summed association of all three short-chain fatty acids. This is looking then at the others of the propionate, acetate, and butyrate levels. And interestingly enough, the butyrate itself was not significantly elevated, but the other two were. But there is the idea that there's a change, but it doesn't bring it back to normal levels for what we find in the stools of our control patients. So we've, again, showing for the first times here that we can effectively raise the levels of short-chain fatty acids just by therapy alone and blocking virus, but it doesn't bring back the normal levels. So what we're now looking at is actually doing stool fermentation and doing this in vitro to then figure out what are the right kinds of prebiotics we could use directly. And these are model systems that we're looking at patients pre-heart, post-heart, and then controls to take a variety of different prebiotics, both soluble and insoluble, FOS, gar gum, and a mixture, and wheat germ. These are all, again, produced by our carbohydrate chemists, and they're now putting these in vitro models of fermentation with stools from our patients. We're then looking here at the differences with the different soluble and insoluble fibers at the butyrate levels. We find different fibers cause different levels. For instance, in the HIV positive, FOS seems to have the best effect, where guar gum has the best effect in our healthy control. So again, we have to look at some precision or personalization of how we look at this. We've then taken and then began to look at these same kind of approaches with John Carn in his models, actually, and shown that we can take the supernatants from these in vitro fermentation reactions where we know have high levels of butyrate and show reactivation of HIV. So we're now doing this and purifying out the molecules to work at the question of restoring back the butyrate levels in vivo and asking the additional question related to the viral reservoir reactivation. Can this increase in butyrate also lead as an HDAC inhibitor reactivation of latent viral replication? This is studies I'll then point to that are also ongoing by Ma Samsok's group, who's really led a lot of the effort in fecal transplants in HIV patients. And this is some of his newest data. Actually, he provided it to me and is not yet published. They've done a phase 1 study that has been published where they did HIV-positive ART-suppressed patients without C. difficile, and they delivered then the fecal transplant by colonoscopy. This was published in Gut Microbes in 2017. So this was the first of the studies to actually look at fecal transplantation in HIV patients. And its most effective means, again, for dysbiosis, especially in C. diff. The phase 2 study that I'll show you a little bit of data on today is in 12 participants, again, HIV positive, ART suppressed, no C. diff, delivered by colonoscopy and by pills, which are known as poopsicles. So this is the new approach of giving back the actual oral fecal transplants instead of doing it by colonoscopy, again, and they have controlled patients. And so the factors affecting the microbiome include the infection, diet, probiotics, stool transplants, and antibiotics. And so in the initial study where they saw very little take of the fecal transplant, what they added in this was the addition of antibiotic treatment with Cipro and Flagyl in these patients was the antibiotic treatment used for 5 days to then preceding the fecal microbiome transplant. Again, and then they did sigmoidoscopies and collected stools, plasma, and PBMCs. I'll show you some of the, and these are the characteristics of the patients with fairly good CD4 counts and also just gives their age range, which are, again, fairly much older patients as well. What this is showing you is that there were strong shifts towards donor profiles, showing you then that the microbiome shift back to what the donor was to give you then an actual reconstitution. But that varied by recipient. So some recipients showed a better shift in terms of other recipients here. So again, variable results in this second study where antibiotic treatment was done. This is just showing the antibiotic conditioning on engraftment. There was, in terms of this phase 1 and then the phase 2 study with antibiotics, maybe a slight bit better engraftment, but again, not a complete engraftment. So these are very early studies, but they're continuing on, and this may give us some other insights in terms of whether or not, and I've talked with Ma about looking further at the functional role, especially, as I mentioned, with butyrate-producing bacteria and the reduction of the bacteria that are involved in driving the kynurenine and the tryptophan ratio, which is more immune suppressive, and the indolamine 2,3 dioxygenase enzyme made by the bacteria, to see if you're actually reducing those and increasing butyrate producers, which is, that work is still ongoing. So where this work really sits today is that FMT is safe and well tolerated. Engraftment appears to be limited without antibiotics. So again, at least antibiotics are necessary but not really sufficient. And this is first studies. No one else is really doing this except his group at UCSF, so I think it's really beginning to understand more about this. The antibiotics did not have a significant long-term effects, as I mentioned, in terms of the communities. Very low alpha diversity may impact the degree of engraftment, and oral delivery may increase engraftment, which I mentioned did better than with the actual GI, and perhaps the small bowel community effects of the long-term colonic community if you're giving it orally. And then how can engraftment be improved is more altering the antibiotics themselves and perhaps the frequency. So all this is ongoing, but I just show this as an alternative approach that at least is being used clinically or experimentally in the field. The other thing that we're producing and thinking about from the aging perspective is how to look at the approaches of reducing inflammation. And here's where we're learning a lot from the aging community. We're looking at targeting new therapies. Everybody loves the tree. I was actually part of this review with Amy Justice where we published the tree. So if anybody's looking where this tree that Peter Hunt designed is published, it's actually in our paper with Amy Justice and JID in 2018. And what we're now focused on is looking at what might be targets like IL-1, JAK-STAT have been looked at, IDO. I'll talk more about metformin because that's in the context of both aging and HIV. And we're also starting GLP-1 agonist semaglutide studies actually in the ACTG as well. So we are looking at these and we do have therapeutic trials underway. One of the fields of aging that I want to bring to this HIV community, or maybe some new concepts you've not been familiar with, that of SASP, which is the senescent-associated secretory phenotype. So I'm not sure if you've heard of that. SASP phenotype occurs in aging. And then senolytics are another class of drugs that are being used in the aging world to actually block or reduce the anti-apoptotic effects. And there are a variety of these drugs that are being looked at. I first of all should point out JAK inhibitors. Metformin is a SASP inhibitor as an mTOR inhibitor, and then NF-kappa B inhibitors. So these are all being looked at in the aging world. And there's a large 3,000 patient trial that starts next month in aging patients with metformin to look at outcomes, clinical outcomes of cardiovascular disease, cancer, and neurocognitive outcomes. The senolytics are also being used, and I'll show you more data around the first human trial with dasatinib and quercetin. ABT-263 is actually navitoclax. It's a BCL2 inhibitor used in cancer. So again, we're looking at BCL2 inhibitors here in the senolytics field. Dasatinib, which I'll point out in a moment, is a tyrosine kinase inhibitor used to treat CML patients, also to drive senolytics, and looking at others like fisetin and HSP90 inhibitors that are being done. So all of these may have differential effects in terms of inhibiting this inflammatory phenotype, killing of senescent cells, which may or may not be harmful, and we are looking at this is being done in the aging community, but we're also moving some of these drugs in vitro, and there is a dasatinib study already started in Spain to look at the role of dasatinib as a latency reversing agent in HIV patients. So these same drugs, it's interesting now, the whole aging field of senolytics, which is the hottest topic. I met with one of the NIA program officers, and the hardest topic at the NIA today is the senolytics. There's just huge interest in this whole field of aging, and these new drug classes is where that is going. So this may give us some good opportunity to cross borders with our aging colleagues to look at this. So as I mentioned, dasatinib and quercetin, senescent cells have an increased expression of pro-survival genes, BCL2, consistent with their resistance to apoptosis. Dasatinib, which is a tyrosine kinase inhibitor licensed for CML, and quercetin, which is a flavonoid. You can go online today, anybody. I had colleagues do this at the last time I presented at ACTG and ordered this from Amazon for themselves. So you can order it from Amazon today if you'd like to take your quercetin, and I do actually know people who've done that. Target pro-survival factors, selectively killing senescent cells, but are each more effective together again, and a combination form of synergy that's more effective in removing these cells. So this is the first study published by Jamie Justice from Wake Forest, was published in Lancet just end of last year, senolytics in idiopathic pulmonary fibrosis. And this is the first in human study using these two drugs together, this was, again, was selected using a hypothesis-driven discovery approach. They're the same agent known to kill them, and there was already mouse studies shown that this worked in a Nature Medicine paper published last year by Ming Zhu, and then idiopathic pulmonary fibrosis was studied as it occurs in late life. Biological senescence is a key driver in this disease and generally fatal within 4 years of those over 60. And they were able to show, interestingly enough in this study, that there was increase in gait speed. Physical function measures significantly improved in these patients with a 12-week trial. So this is the actual first study in humans to show an actual functional effect of these drugs in aging. So now we're thinking about these same drugs together. We're looking at them in vitro models and talking within our ACTG group with the aging community about the role of these drugs in future studies. The last drug I'll talk about and end my talk here is metformin. We're also beginning studies, large trials of thinking about metformin as the next drug, which is also, as I said, a SASP inhibitor. We're beginning to think about this particular drug. I know has had an interest with many people, but it looks like in the aging world, this is like the hottest drug coming out. And there were at least 4 abstracts at CROI this year on metformin and its potential role in HIV, both from its effect on driving down inflammation and in aging as a SASP inhibitor, but also potentially in latency reversal. And groups like Dorothy Lewis's group in Houston have been actually looking at this in latency reversal. It has effects, as you know, it's a drug used routinely in our diabetic patients, increasing in glucose metabolism, endothelial function, may have neurologic effects and important in improving gut dysbiosis. So there's a lot of potential for these drugs going forward. A lot of these drugs, as I said, are already in clinical trials in aging. So it's not, and these are then moved forward already. Metformin starting in another month. The D&Q study already completed, and there are a whole range of newer senolytics coming down, and we're beginning to talk with the field, with the group that are actually funded by NIA to develop the whole senolytic pathway. So we're now having crosstalk between the HIV and aging community around this. So what I think what I'd like to summarize then for my talk is that there's a lot of potential. I started with the microbiome. I've given you a broader view of life, but I think we can learn a lot from our colleagues across the aisles in other fields, and I think have a lot of potential for going forward in novel therapeutics in the future. I'll just thank my collaborators. As I mentioned, work with Steph Dillon, Carol Wilson, work on the cardiovascular metabolic work here at Albert Einstein, with works at the University of West Indies on the glucose metabolism. I mentioned Moss Samsuk's work at UCSF, and then Mohammed Abdul Monson's work from the glycomics. I'd like to thank you for your attention, and happy to answer any questions.
Hello, very nice talk. I'd like to know if, I don't know, considering that heart itself could change some microbiome factors, I'd like to know what's the possible consequence of PrEP in these individuals who opt for this prophylaxis.
So that's a great question. Michael Dubé actually published a short report in Science Translational Medicine this year on the role of PrEP, where he did not show any significant changes of PrEP on the microbiome. Sandra Pinto-Cardosa in Mexico City at CNE actually has done work and written up and shown effects. And she's actually going to give a talk this year. Again, some shameless advertisement for our HIV and microbiome meeting that we hold every year at the NIH will be held in October, and Sandra will be giving a talk this year on that very topic and some of the current data they've been generating at CNA in Mexico on the effects of heart. And they do find effects of protease inhibitors, especially changes in the microbiome. But I think that in the PrEP world, we're doing some studies with Jeff Jacobson at Temple, and I'm sure there are many other groups who are looking at the role of PrEP in the microbiome, and I think there will be more data presented in the next coming years.
Hi, Alan. I was wondering do you believe, given that HIV and aging have independent effects on microbiome, do you believe that the probiotic, prebiotic, postbiotic interventions are going to be more likely or less likely to benefit HIV-infected older patients?
Yeah, that's a great question. I mean, we're still in this proof of concept stage, and our recent probiotic Visbiome study, again, based on Jason Brenchley's JCI paper, showing significant effects in the monkey model with Visbiome and SIV. We did not see any real significant effects in our clinical trial in the ACTG. And though in our patients, we're not necessarily all older patients, but I think it's going to be hard to see what we can do. I think if we can understand targeted microbiome, which is what I was pointing out in aging, perhaps, and what we're trying to do now with other collaborators is using culturomics as an approach to actually culture directly the specific bacteria that are found in HIV and aging patients to try to figure out what is the metabolic profile, and then use more targeted approaches. I would agree, these were nice proof of concept trials, but they haven't yielded very much fruit to date. So I'm not as positive about them. I think they're nice proof of concepts, but in the long run, they probably will not give us a good effect in a broad sense. I think we're going to have to be much more targeted, much more focused around particular metabolites, and we are doing those studies now and using those particular approaches.
What's the impact of metformin on dysbiosis, and what are the trials being conducted? You mentioned a 3,000 persons trial, and what's it about exactly?
Well, we're still, that's a good question. What we're doing now is designing the trial. A lot of it may be related, the metformin trial and HIV, we're actually looking at perhaps more latency reversal questions, since that may be more interesting. We are trying to figure out what are the inflammatory markers, and since this is in our ACTG studies, there have been questions about many of our trials, and Michael might want to comment, and he's participated, is we haven't seen positive results. It may be the patient selection, it may be other reasons. Many of our approaches for driving down inflammation, similar to what might be done with metformin, haven't been successful. So we're still in that very early design stage. We have a concept or a proposal we're sort of thinking through, and what are the right outcomes. I think that is still a critical question, and what are the right patients in terms of the baseline alterations. But I'd like maybe Michael could comment, since he's thought about it as well. Yes.
Wonderful talk. Thank you very much. And you introduced several drugs for therapy that affect gut microbiome. And my question is there the effect of those drugs on microbiome in the other sites, for example, oral microbiomes or so on?
Yeah, that's a great question, Tetsuro. A lot of this talk focused on the role of the gut microbiome. There's been more limited work looking. We are starting studies on oral microbiome and linking oral, gut, and vaginal microbiome in women, and trying to look across different sites in our HIV populations to get at that, because there's been very few studies that take multiple sites to look at that, and we're just now at that beginning phases of doing that. But we are planning those studies, and I really can't speak to the idea. We don't have a lot of data yet, and that's just the early phases. Michael?
So, Alan, in response to your question, if our studies don't give us the answers we want, then the obvious thing is to blame our patients for their failure. Right. But I was going to ask you, you're talking about senescence and aging and the relationship to the microbiome. Our cells, when they divide multiple times, they get tired. They can't do it forever. Is there any analogous thing when it comes to bacteria or fungi? If they undergo multiple rounds of division, which they do much more frequently than we do, do they tire out? Do they age out, or are they just good forever?
That's a great question. I'm sure somebody who's more attuned in the mic, maybe somebody here who's looked at that question. I don't know if you get senescent microbiome as well. That's a great question, because they could also, like our cells, and it's interesting, the cells that become senescent and become resistant to apoptosis in aging, it's the same thing happens when we activate our cells with HIV, and the kick in that when we activate latent virus, you upregulate BCL2 expression, and so that's why a lot of these agents are there. But I know we've argued that happens, and some of the stuff that you've seen is not the same data. Yes, Rafiq.
So I'll add one comment and one word of caution. I mean, the first comment is I think related to the fact that maybe you're not seeing results in your clinical trials. And you probably know our work, that I mean, you cannot lump all the subjects into one big lump, and I think what we clearly see, and we validated this by 10 different approaches is the fact that in our subjects, I mean, these subjects are very heterogeneous, and even that their microbiome is very different. So in subjects, we see in some subjects a very inverse relationship between proteobacteria and butyrates, and maybe I think if you, or firmicutes. So if you probably target at baseline, I mean, if you do your screening based on the different microbiota, and now you target, I mean, those subjects with the appropriate drug, I mean, and I think, I mean, that would be for you a way by which you might be able to generate a better result. The second one, which is which maybe maybe you're looking at very different things, you know, but the data we have on butyrates are exactly the opposite of the data you have. And I just want to remind you that, I mean, what Sasha Rudensky and Hiroshi Hono showed is that butyrates trigger Tregs. And if you butyrates trigger Tregs, Tregs are going to produce TGF-beta and completely inhibit HIV replication. So, and we have tons of data where we show, in fact, that when you take butyrates, you completely reproduce that same pathway, and you add your Tregs, I mean, to cells that replicate HIV, and you completely inhibit HIV replication. So and even if you take butyrates and you add them to T cells, you induce senescence, and that's the work that's published by Judy Campisi in Cell and two other groups more recently. So butyrates trigger senescence, and if you trigger senescence, you're not going to reactivate HIV. So I mean, I really would caution you about looking at the way you analyze your data, because
there's always
they're going opposite way to what the literature has published recently.
Yeah, I mean, we've looked at, again, the butyrate in terms of a lot of work has been looked at butyrate in other systems, in cancer, in IBD, and all of it's being used to enhance gut barrier integrity. We haven't looked at it directly in the aging population, so that's a different question, and I would agree that's something that probably needs to be looked at, because there's always going to be a plus and a minus effects of some of these effects. So where you're driving, and clearly, as you said, butyrate is going to drive a Treg population, and that's what helps to enhance that. But there's also the gut barrier function, and in other conditions, they're using butyrate therapeutically as well to treat other conditions, including CNS-directed conditions and allergic conditions. So it's being used therapeutically. What that does...
As an anti-inflammatory drug, not as a pro-inflammatory drug.
Right, as an anti-inflammatory effect. So, yeah, I think it's context-specific, and clearly, as you point out, if there's a role in aging and using it, we need to be careful about how we look at it in an older population as well.
You know, redesign maybe your clinical trial. If you're designing your clinical trial, try to classify them based on their microbiome before you even test your drug, because I mean, you might hit the wrong population. If you classify them prior to initiate your trial, you might maybe see much better result than what you see now.
Sure. The pre-screening questions are very relevant, and we've talked about that many times to try to do that. I think it's, in some of our large trials, very difficult to do, but in single-site trials, I think it's something we can do to get proof of concept. So I think that's a great idea. Thank you.
So thank you. I think we should move for the next talk. Thank you very much.
Thank you.